Hi, I'll just wait for 2 minutes maybe and then I'll start the session. Just wait for one more minute to see if anyone else joins, or I'll just start sharing my screen. Okay. I'll start in case someone else joins. Okay, hi, I am Paulimi. I'm a PhD student at Indian Institute of Science, and I'm a course member for this course, which is called Basics of Biology, offered by NPTEL. And so, what I've decided is in this problem solving session, I'll start with going over the topics that has been covered in this week. So, for example, this week I am covering the topics that was covered in the previous 2 weeks. So, I just go over the main topics, and you've got assignments to do. So, we'll also go over questions based on these topics from previous years, and we'll try to solve them. And if you have any problems in any of the topics or any of the questions that's coming up, and you can ask me that. So I'll just share my screen. Can you see my screen? Could you confirm that? Sure. Okay, thank you. Okay, so like I mentioned that we are doing basics of biology course. And so like I mentioned that I'll go over the topics that were covered. So I'll start with the basics. So what is biology? Biology is bios and logos, is made up of these two terms. Bios means living organisms, logos means to study. So it means in taking in together, it means the study of living organisms and their vital processes. And it has two major divisions: botany, which is study of plants, and zoology, which is study of animals. So how do you define a living organism which is like different from something which is non-living? First of all, it has to be made of cells, and these have complex organization, which means that they will be constituted of a structural component and a functional component, and the functional component will be used to obtain and use energy and metabolism, which will help them grow and develop. They'll be able to reproduce and movement, but the exception being plants, because plants do not move, but they are still living organisms, and they can evolve or adapt to their environment, and they are sensitive and respond to the environment or stimuli. So this is one something like you can change according to what you are perceiving. So that makes you a living organism. And because we're talking about biology, it is important to know how did the biology as a field got a stronghold. So these are the scientists who, with their different contribution, made sure that this field progresses from the 18th century or the 16th century. So we have Aristotle, who is known for whose major contribution has been the classification of biota. And we have Galen, who was the first one to perform dissections. We have Andreas Vesalius, who was the first person to do comparative anatomy, and we'll understand why comparative anatomy is such an important field or such an important topic for biology as a field. We have Marcelo Malfigi, upon whom the Malpighian tubules from the insect body has been named, and he was founder of microscopic anatomy. William Harvey, who was responsible to show that the heart pumps blood and these blood is involved in circulation. And most importantly, Antoine Leeuwenhoek, who was the first to use microscope and with which he discovered microorganisms such as protozoans, which will come across that is a class that is used in classification of organisms. And last but not the least, Charles Darwin, who has written the book on the origin of species, who has given us the concept of natural selection, which underlies most of the evolutionary processes that we see in our nature. So, like I mentioned, that biological systems are complex in nature. So, just a second. So sorry. Yeah. So, like I was mentioning, that biological systems are complex and they are also very diverse. So, we have different parameters and we can see how diverse these biological systems are. So, for example, shape. You have prokaryotes who are ranging from micrometers to eukaryotes whose cells can be very big. Then we have size of the whole organism. So a virus can be 100 millimeter in size to we have blue whales, an organism who was 70 to 80 feet long. Spatial, it means where are they distributed, where they reside, what is their habitat. They could be in water, they could be in land, they could be residing on top of trees, inside the burrows, on the land. There are multiple habitats or niches that are available for these organisms to survive. And then biological activity. So they are not only present, like I mentioned, they're not only present on water and land, they could be in caves. And if they are in caves, so they are very much adapted to cold conditions. If they are present in hot spring, they are adapted to hot conditions or highly acidic condition or salt condition. So all these organisms who are living in different habitats, they have all evolved to be there, and they have all evolved adaptations to survive the best there. So that's how biological diversity is enhanced. And so, so like I had mentioned that we divide our organisms into several kingdoms, so that and on the basis of characteristics just present amongst them, which makes us understand how the evolution has taken place and how the organisms have continued to gain complexity over the period of time. So we have some certain set of properties, and we can define five kingdoms based on those taken together. So for example, cell type. So we the first five kingdoms in overall we can say are Monera, Protista, Fungi, Plantae, Animalia. Now, how do we define them? We have cell type. We can see that Monera is prokaryotes, which means that it will not have a nuclear membrane, it will not have a cell membrane, and eukaryotic, which will have proper nuclear membrane, and then we have proper organelles and a cell membrane and a cell wall are all present in these kingdoms. Then we have cell organization. So, what is the basic unit of the cell? Are these only one cell which makes the whole organism, or are these different cells coming together that make up the complex? So, we have unicellular completely for Monera and Protista. In fungi, we have both the forms. It can be unicellular, that is one cell, or it can be multiple cells coming together and forming the organism. And for plant and animal, they are multicellular organisms. And then I mentioned about cell wall. So it's present in Monera, it is present in Protista as well. The absence which we observe in Protista are some of the exceptions or some of the unique characteristics of those members. And then these are present in Plantae and Animalia, but Plantae and fungi, but not in Animalia. Animalia will not have a cell wall; it will have a cell membrane. Nutritional class is how do they obtain the food. So you have to obtain your food and use that energy to survive. So either you make your own food or you use food that is available to you in the nature and you just use it for gaining energy. So on the basis of that, we have phototrophic, heterotrophic, and chemoautotrophic. And so phototrophic means using light and using raw material, you are making your own source of energy. Autotrophic, it means you are making your own source of energy, but using chemicals and raw materials. So Monera can have all three, phototrophic, heterotrophic, and chemoautotrophs. Protista can have heterotrophic and photoautotrophic. Fungi are heterotrophic. Heterotrophic means they will be using some other material which is already made by some other components or other organisms as their raw material for energy. Similarly to animalia. And plantae, as you know, plants are able to synthesize their own food by the presence of chlorophyll and the presence of photosynthesis. Yeah, so sorry. Okay, one thing that I forgot to mention was if you have any doubt in any of the slide, do stop me and ask because I can't see that if you're raising your hand. So please just pitch in and ask your questions. Okay, so next is how are they like how are they using those food? So we know that they have prepared their food, but how are they using it? Are they taking in via absorption or are they actually ingesting it via some mechanism of engulfing or taking it in via some procedure? So if we have Monera, which is bacteria, they're generally absorptive. They absorb via their cell wall. And for Protista, you have absorptive or you also have ingestive where they form a vacuole around the food source and get it in. For fungi, you have absorptive. Plantae also absorptive because you know that plants with their roots are able to take the nutrients from the soil and then use it up for their food sources. And animalia, as you know, they are heterotrophs and they are ingestive in nature. And now we come to motility. Now I mentioned that for living organism they have to be motile, but that doesn't mean that it has to move. As in, a movement means moving from one place to other, but it doesn't restrict an organism from being a living organism. So what I mean is, for example, bacteria, they are some of them are motile, some of them are non-motile, but they have these other components which makes them use energy, survive, reproduce most importantly. So we also use motility as a characteristic to define a particular kingdom. So we have Monera, Protista as being motile or some of them non-motile, fungi and plantae being non-motile, animalia are mostly motile. Now we'll go each of these kingdom one by one. I'll just briefly touch over this kingdom because as I said, I'm just going over the topics that have been covered in the lecture notes. It's just to give you an overview of whatever you have learned in this week. So Monera are unicellular organisms. They consist of 70S ribosomes. They don't have well-defined organelles like mitochondria, lysosome, plastids. As I mentioned, they reproduce for sure, and they do it by asexually, which is by binary fission, by splitting up into two. Budding, which involves a formation of a budding bud, an outgrowth, and that outgrowth splits off and forms as a new organism. Flagellum, if as it's a locomotory organ, if it is motile, and Monera or bacteria are also defined into three categories. There can be archaebacteria, which are the ones that survive in very harsh conditions. Eubacteria are the ones that we see in normal conditions, and cyanobacteria, which have the chlorophyll, which helps them to synthesize their own food. Coming to Protista, they are also of three types. They are protozoa, slime molds, and chrysophyte, dinoflagellates, and euglenoids. So protozoa again are of four different types. They can be amoeboid protozoans. These have pseudopodia. So the pseudopodia is the mode by which they engulf their food. Then they have flagellated. So this flagella is what defines them. This flagella also helps them in movement. So we have euglena here, for example. They can be free living or parasite, means they can survive on their own or they need a host to survive. Then we have ciliated. So in these protozoans, for movement, they use cilia. These cilia are small hair-like structures all over the body. And these are aquatic in nature. And then we have sporozoans. So what are these sporozoans? They will have a spore-like structure among their life cycle. And that spore-like structure is something like this, where they will be in a cell in a cell, and these all these components are shaped like this together, and then they will break off and form their trophozoite, which is carried on in host. So for example, plasmodium vivax. And the other classification is for slime molds. Now these have many nuclei, not many cells, but many nuclei. So these are multinucleated cells. So they are all the cells together, but then all the boundaries have matched, like bordered, have formed conjoined, and the cell nucleus are all retained together. So these are aggregates. So just imagine few of the cells coming together. Their boundaries overlying with each other, mixing, gelling with each other, and they're all becoming one single entity characterized by presence of many nucleus. And then we have chrysophytes, dinoflagellated nucleonoids. These can be single-celled or multicellular. And the most important thing is they are photosynthetic and found in freshwater sources. And they are also very important from an economic perspective because they are used for the extraction of oil from water. So they have these other characteristics which make them very unique. Then we have fungi. So these fungi are unicellular and filamentous. They reproduce by means of spores. Spores means they will have these will be small little cells which when are able to move to a new place or on the soil, they can germinate and they can give rise to the whole organism. And they exhibit the phenomenon of alternation of generation. Basically what that means is that they can be gametophyte or sporophyte, and because they are heterotroph in nature, they store their food in the form of soil because they don't produce it. So if they get this the food, they have to store it so that they can use it later for purpose. They can reproduce via sexual or asexual method, and they can survive as parasite, and they can also or infect the host, or they can also like they are parasitic in nature, so they are not free living. And these fungi also, they are like they're divided on the basis of what kind of food they are taken and what kind of spores they are forming. So if they are saprophyte, they can be saprophytic, they can be parasitic or symbiotic. So parasitic is a interaction where you feed on the host and the host is not receiving any gain from your interaction. But symbiotic is where you are working with the host also is an advantage and you are also an advantage. So symbiotic and parasitic are defined by the the interacting members and how they are getting advantage out of this interaction. And based on spore formation, so what do we mean by spore formation is how do these spores are like released into the environment? Are they in a disc-like shape, or are there single spores that are coming out? So on the basis of that, you can have zygomycetes, ascomycetes, basidiomycetes, and deuteromycetes. And then we come to plantae. Plantae, as I mentioned, that they are non-motile, they make their own food, and so they are called autotrophs. They asexually reproduce by vegetative propagation, or they can also reproduce sexually. And they are autotrophs, they synthesize their own food due to the presence of chlorophyll, which are photosynthetic pigments, and they are present in a special organelle called the plastid. And similarly, they have different organelles for anchorage, for reproduction, for support, and photosynthesis. And then coming to Animalia, which is the largest kingdom of all, which do not have cell wall, do not have chlorophyll, so they are heterotrophs in nature. And then we have some organisms which are non-canonical. They don't have these special these characteristics that can make them fit into one of these five kingdoms. So they are left on their own and they are treated on their own. So for example, viruses. These are inert crystalline structure outside. So if they are outside the living cell, they are inert. They don't reproduce. But the moment they find a host to latch on, they can infect this host, get into the host machinery, and then reproduce themselves and get. That's exactly what the purpose is of a living organism, to reproduce. So these are inert crystalline structure. They but they once they infect a cell, they can take over the machinery of the host cell to replicate themselves. And then they are also formed of nuclear material that could be RNA or DNA, that could be single stranded DNA or double stranded DNA, similarly single stranded RNA or double stranded RNA. And then we have viroids, which are smaller than viruses. So remember, virus itself is very small in nature. Viroids are even smaller than them. And these, so they can be called as free RNA, which do not have the viral protein code. And then we have lichens. So lichens is an example of symbiosis where we have the beneficial interaction between the algae and the fungi. Fungi we already covered in the last slide. So, like I mentioned, that they are complementary to each other, and how is that? So, as you know, that fungi is heterotrophic and algae is autotrophic. So, algae synthesizes food, and fungi uses that food. But what does algae do? Like fungi does that. In fungi, what is the beneficial component for algae in this interaction? Fungi provide them the shelter, and because they are absorbing minerals, algae can use the same mineral for using raw as a raw material to synthesize their food. And one very important Feature about lichens is they are good pollution indicators. They will not grow if there is pollution in the environment. So if you see a lichen growth somewhere, you know that environment is pollution free. That's why they're very important, and that's why they have their own importance in this classification system. Okay, now we talked about kingdom, but I also mentioned that these organisms are very complex, and there are so many organisms that you just can't use these five characteristics to classify them. So once we have defined a kingdom, this is particularly for plantain animal mammalia. So you have a kingdom saying animalia, but that doesn't capture every organism and every characteristic that is present in them. So what you do is you further classify them into several other small sections taking common characteristics and then you go to the basic unit. So you start with kingdom, which is the broadest of all, move towards phylum, which has again some characteristics common in them. Then you go to class, then you have order, then you have family, genus, and species. Now remember, when you go from kingdom to phylum, that is just one step. But when you go to phylum to class, in between you can have several subphylums as well. Those subphylums are even further characteristic taken together. Similarly, when you go from class to order, you can have subclasses. When you go to in order to family, you can have infra order or suborder. The infra order is below orders, below suborder. Then you have family to genus. Family and genus, you have in between you will have tribe or subfamily, again some characteristics taken together, and then genus and species. If you want to remember this as a mnemonic means if you want to remember how is this classification uh, system goes on, just take the first letter of these ranks and make a mnemonic out of it. So what I do generally is I remember it by saying keep ponds clean or else frog gets sick. So keep for kingdom, ponds for phylum, clean for class, or for order, frog for family, get for genus, and sick for species. So that's how I remember the ranks in which they are classified. And it is important because when you define an organism, you need to know where it is placed in this pyramid so that you can properly define them. And with if you just tell them it tells something as saying that they are diptera, for example insect, then you know that what are the characteristics of this organism of this insect. For example, also in like snakes, so if you say them as a viperid have different characteristics than Lapid means, for example, their fangs is different, their scales are different. So if you just say a snake is viperine, you know what all features can be in that snake. And this system that we are using for classifying organism was given by Carl Linnaeus. And so what is this? This is a method of systematically arranging the different species of living organism into closely related groups. And like I mentioned, this is scientific ordering of organism into different taxonomic group, which is based on several characteristics like morphological, anatomical, and evolutionary. So what are the criteria that we use for animal classification? First is level of organization. Are they cells? Have they formed tissue? Tissue means all the cells coming together, and then each aggregate of cells take up different functions to perform. So they're tissue. Organ is when these tissues come together and then they together perform a particular function. Then we have organ systems, and then finally we have organisms. And then we have symmetry. Symmetry is how does that body like how is it organized? So what I mean is they can bilateral. So you have this one line of symmetry across the organism body. You have radial symmetry. That means there are multiple planes for equal division. And you have spherical. So there's a spherical body and you have these different planes to divide them. Then there are germ layers. We'll come to germ layers where we mean how is the, what are the different germinal layers that they have. So if they are two, which means they have only endoderm and ectoderm, they are diploblastic. If they are triploblastic, that means they have a mesoderm in between the endoderm and the ectoderm. And these are important because each of these layers give rise to different parts of the organism. And then you have notochord, which is, so you have a nerve cord and you have this basal layer beneath the nerve cord, which is also a characteristic which can be used for classifying. And then you have segmentation. So if your body has segmentation, that's first step. And then how are those segments structured. So are they in the thorax, are they in the head, are they present everywhere, or are they in the abdomen? So based on that also you can define an organism. So I'll just go here. I see lots of new joinees. So I'll just say, repeat that if you have any doubt on any of the slide, please just unmute yourself and ask, because I'm not able to see if you have raised your hands. Okay. So, based on all the characteristics that we have read now, which are used to define classes of organisms, we'll first divide kingdom Animalia into further subclassifications using the characteristics that we have just gone over. So, if you just see this map, what it means is, just 2 minutes. Yeah. So this is the kingdom Animalia. We use first the level of organization, whether it is cellular or tissue level, and then we go to symmetry, and then we go to body cavity. So what is body cavity is a fluid filled cavity which arises out of which arises out of the mesoderm. So you have your ectoderm, you have your endoderm, and in between you have mesoderm, and then mesoderm will again split off to give something called a coelom, which is a fluid filled cavity basically, which is important for giving structural stability to the organism. So the first group is porifera. Then, based on tissue level of organization, we move further, and then we divide them according to symmetry. On the basis of radial, we have Coelenterata and Ctenophora. And on the basis of bilateral, we then go forward and divide them, saying that if they have coelom or not. So coelom, true coelom will come from mesoderm. Pseudocoelom will have just, so imagine that it is a fluid cavity. So if it is circle, the coelom also has to be circle. But if it is pseudocoelom, it will be just in patches, like here, here. So if you just imagine this to be the mesoderm layer, and then the coelom will be like this, some concentric circles here. But if it has true coelom, the mesoderm itself will split into proper circle to give true coeloms. So based on that coeloms, you have platyhelminths who are acoelomate, you have esculpins who are pseudocoelomates, and then you have coelomates which are annelida, arthropoda, mollusca, gynodermata, and hemichordata, and chordata will come across little later. So chordata is again, as I had mentioned, there could, before going to the level of class, you have subphylums. So it has three subphylum. First is Urochordata, Cephalochordata, and these taken together are also called Protochordata. And the basis of dividing them is the presence of vertebral column. So if you have notochord, yes, that's why you are chordate. But do you have a vertebrate column or not? If you have a vertebrate column, you are vertebrata. And then after you are a vertebrate, what you first see, do you have a jaw? Jaw is important because this jaw was not here until now, because jaw gives you a structure for mouth. All the previous organisms could use suction as a mode of feeding, or absorption as a mode of feeding. But here we have come to a structural form where we have vertebrata. So we have vertebral column, we have coelom, we have gut. So we have all these organisms, and now we are coming to the next advanced structure, which is the jaw. Now in this also, if there is no jaw, they are called cyclostomata. So for example, lamprey and hagfish. So if you just see here, this is a lamprey. They will have this particular disc-like structure from where they can suck their food sources. And then if you have jaw, then you divide them on the basis of further characteristics. So if you have fins, then you're fish. But do you know that their fish also can be divided into two types, which is on the basis of bones. If you have bones, then you're ostrich thighs. If you don't have bones, then you are chondric thighs. So the previous one was shark, so it is chondric thighs. This one is a normal fish that you see in any like museum or aquarium. So that is ostrich thighs. And then you have, so fins as a mode of locomotion makes you fish. If you have limbs as a mode of locomotion, you are tetrapod. And in tetrapod, basic mode of dividing these organisms are how they where they reside. So if you are on both land and water, you are an amphibian. If you are on land, you are reptile. If you have feather and you can fly, you are ape. And if you are able to survive anywhere, and you are even more advanced than A, but you lack your wings, then you are mammal. Now, moving on to plant classification, which is again a very, very vast kingdom which has multiple organisms. So, you need to divide them further. So, what characteristics do you use? You use plant body, you use vascular system, whether they have xylem and phloem or both or single, and seed formations. So, how are the seeds? First of all, seeds are formed, yes, but are there fruit formation? If the seed is formed, are they naked? Are they winged? What type of seeds are formed? So this is a broad classification of plants. So you see, first thing is whether the plant produces seeds or not. If they don't produce seed, then how is the body type? Does it have everything? If it has everything, that is root, stem, and leaf, then they are ferns. If they have no roots, no stems, or no leaves, algae, and fungi. See, these are two different kingdoms coming together to form a symbiotic organization, which was called the lichen. And if you have roots, stem, and leaf like structure, so remember, you have root, you have stem, but you have something like leaf but not true leaves, so then you are mosses. But if you have true leaves but you are not producing any seed, then you are fern. If you are producing seed, do the seeds also, like, do the plant also bear flower with them? If they bear flowers, they are called angiosperms. If they don't bear in flower, they are called gymnosperms. For example, conifers that you see in the high mountain areas. And then if you are angiosperm, how is your seeds? Does it have a single cotyledon, or does it have two cotyledons? Two cotyledons are basically how do, when you germinate them, how do they start with their germination layer? Do they have one outgrowth, or do they have two coverings? So if you have one, you are called monocots. If you have two, you are called dicots or dicotyledons. So this was all about classification that was covered in the first part of the classes. We also covered origin of life and evolution. And this is equally important because to understand all this diversity, you first need to appreciate the fact that all this diversity are harbored only in Earth as of now that we know. Let me mention that. But yeah, so all of these diversity is harbored in earth, but first the earth has to have come about somehow. The earth needs to form so that it gives the amenable environment for all these organisms to develop. And then all these organisms, did they come from one single organism, or did they come from multiple like different events? You need to understand to appreciate the nature of evolution going on. So first, like I mentioned, we'll start with how the earth was formed. And then in this earth, how did it become from like a solid land mass to a land which is, or a planet which is filled with living organisms. So we'll try to cover that trajectory in the coming few slides. So first thing is, earth and other planets in the solar system came to existence around 4 to 5 billion years ago. And when it started, earth, it started with as a land, like, it had two components. It had the solid mass lithosphere and the surrounding gaseous environment. And so you can imagine that when it started, or when it was first formed, it was a very hot, the temperatures was very high. So when you have this hot bubbling land mass, and you have very high temperature, you cannot have any chemical reaction going on. But once the temperature started to cool down, there was a layer which is also called hydrosphere, which was formed, because when the temperature started cooling, condensation started happening, and you had these water bodies forming. So you had a layer of hydrosphere as well. So that was how, in a very brief, how I have mentioned. But how the Earth came about to be, or how the life came about on Earth, is also like, there are major theories that may like answer this. And it's important to go over each of these so that we appreciate the actual process. So the first is theory of special creation, where it says that all living organisms were created similarly, and there are no differences in their appearances, and they were created the way they exist, and there is no evolution. So what it means is, there was one this defined creation who had just put on all these organisms the way they are on the planet, and they have remained as such ever since. Now, if that is the case, you would not see adaptations happening. You will not see an organism changing to its environment, and that is why that is one of the drawback of this theory. And other than that, it is just a religious belief, so there were no scientific evidences to support the theory. And you have fossils who demonstrate that biological organism appeared on earth at various times. So basically you have fossils who have given you a timing saying that this has evolved so and so million years ago than this. So if that is the case, that something has appeared before something else, then they all could not have been there at the same time or since the start of the earth. So that's how this theory is not held important. And then we have theory of spontaneous generation or abiogenesis. So this is given by Aristotle, who said that yes, there are living organisms and they are diverse because they can arise out of non-living organisms. So what he did was he had a piece of bread and cheese and he left it to be. And he found that there were maggots which was forming out of these meats. So if you are in the audience, I'll take 10 seconds to think that how could this have generated. I'll give you an answer in the upcoming few slides. How could have this happened? Because I am saying that this does not support spontaneous generation at all. Because he says that this living organism arrived out of non-living organism. But I say no, it is not the case, and why I will justify in the coming few slides. But if you can, I'll just give you 10 seconds to think how could this be? What could have helped the growth of these maggots? So that's what Aristotle mentioned, that this is abiogenesis, wherein there's creation of simple organic compounds from non-living matter. Again, though even though he had proved, it was refuted by further experiments like these. So Reddy's experiment. Now the idea is that you keep your stuff and you should be able to see growth of living organism from this non-living stuff. So what did Reddy did is he took a piece of meat. Now he kept it 3 jars, and I like to highlight something here. This is a very well designed experiment. You refute several grounds of assumptions that you have started with. So you kept the first one. He kept the first one as Aristotle defined, that you have your piece of bread and you keep it open, and then you will see that they have maggots formed. But additionally, you also see that these flies are coming in, and maggots are the younger flies. So that's how you maggots are young flies. So you have maggots seeing here. Now you stop these flies from coming completely. You don't want flies at all, so you put a cork or a parchment here and you stop these flies from coming in completely. So you could say that okay, because the flies did not come, the maggots did not appear here. This is what it looks. But to be completely sure that even if flies come and sit but does not come into contact with the meat, the maggots will not come here. So in the third one, he put a gauze. Now in the gauze, via the gauze, you can have smell coming out. So because of the smell, flies keep coming and sitting on the top of jar, but they do not go onto the meat. But you see here, when they sit here and they're all aggregating here, they start reproducing and they have this maggots formation here. So if maggots is formed here and if maggots is also formed here, it means that the presence of meat is not the character or the feature which is important. Presence of flies with each other is important, and flies are living organisms, so it means that it is not arising out of non-living organisms. Then we have similar experiments by Spallanzani. So what he did is he had a broth. So this could be like it could mean animal broth or a plant broth. He heated it up and then he left the flask open. And then he waited for some time and he saw a growth in this broth. Now, what is the motto of heating it up? Heating of this broth killed all the microorganisms or living organisms present in the broth at the start. So when you are here at this stage, you are completely point zero. You have nothing in this broth, and then you wait. And if you then see a growth, it means it has all come up anew or afresh. So you see this. But then he did something else. He heated, he kept the flask as it is, I mean, with the broth, but he put a cork on top of it. Now, a cork, so there is no air coming in into the flask anymore. and he waited and he saw no growth. So he could deduce that if there is no air, there is no growth, because air brings into contact different microorganisms or organisms into the broth and they start reproducing and growing. But how do you confirm that? You take the cork off. Now, the moment you take the cork off, you again bring air into contact with the broth. And as and when you bring the air into contact with the broth, again there are organisms growing in the broth. So this confirms that you need air, you need some sort of interaction between a living organism and the broth to bring about new organisms. And it is not just the broth that can uh, give rise to new organisms. And the third sector of experiments was by Pasteur. So again, he also said about a life can arise from pre-existing life. So he did the same thing. He applied heat, let the flask sit, and he found no bacteria. Then the most important feature of this experiment, or how his experiment can also be termed, is called the S-shaped flask. So S-shaped flask means that there is this. Just a second. So S-shaped flask. So what he then did was he removed this flask entrance. So he removed the S shape. Now he's did the same thing that was seen in the previous slide. That the broth is now in contact with air, so you see bacteria. And then what he did a further alteration. So while he was heating, he tilted the flask. Now see, when you are heating the flask, you have these parts, these broth which are not in contact with the heat. So you are letting it sit, and then you find organisms growth, because you have some of them surviving here. So these guys have again given rise to new organisms. So you say that life has arise from pre-existing life. So unlike the theory of creation, special creation, where there were no scientific evidence to even refute the theory. For abiogenesis theory, you had scientific evidences to disprove the theory, and that gave rise to biogenesis theory, which says that production of new organisms, new living organisms, comes from pre-existing life. Biogenesis means bio means a life coming out of a life, or a beginning of a life from a life. And this was given by Rudolf Virchow, and it was proven by Pasteur, as we have just seen. And it is also called omnicellular cellular. So a cell is comes from a pre-existing cell. Then the other theory is also called the cosmozoic theory, where they believe that there were these all these organisms that we see. They did not arrive, or they did not evolve on the planet Earth. They were from some other part of the solar system or universe, and they just came to be on this planet by several meteorite attacks. So it was given by Richter and Arrhenius, because they found several meteorites which had uh, amino acid constituents similar to what we find in living organisms here. So the meteorites had those similar composition, which made them think that it was meteorites which got these different organisms onto the planet Earth. So according to this theory, life had reached the planet Earth from other heavenly bodies in the form of highly resistant spores of microorganisms, and these spores are called cosmozoa or panspermia, and because they are preserved in the meteorite, so these spores are able to give rise to different organisms. And these meteorites struck the barren earth to release the cosmozoa, and because of which they germinated on the planet Earth and they diversified. But that is also not true, because of the coming experiment that we'll just see. So now what we actually believe is this chemosynthetic theory, or the modern theory of origin of life, which is also the scientific hypothesis, because there is a proper scientific evidence of how did life came about to be on the planet Earth. So what it means is that first life arose from a collection of chemical substances, and all those had a series of different sources of energy at different point of time on the planet Earth to form different compositions. So we'll just go over it. But the experiment that actually gave rise to this thought was for Miller and Urey experiment, who tried. So they were Operin and Haldane, who had said that the origin of life is biochemical in nature. So they mean that this life, as we see today, as an organism with a particular structure of wings, they did not they were not born with these. They started with simple biochemical components, for example DNA, for example RNA, for example protein. And these proteins is what made up the entire organism. And for testing this, Miller and Urey did an experiment. So what they did is, we'll start with here. So there's a cyclical flow here. So if you see this chamber here, this is called a boiling chamber. Here they started with uh, uh, gases. It's like ammonia, water, and carbon dioxide in the ratio of 2:1, and they had very high temperature. So what they tried to mimic here was the primitive earth. Remember I told you earth started as a very high boiling mass, land mass. So they were burning this temperature to around here 800 degrees centigrade. So this was mimicking the primitive earth conditions. Then what happened, remember, then we told that the conditions on the earth subsided, the temperature went down. So here this the water vapor started condensing because the temperature was kept low here. So as and when these gases who were in boiling chamber here started moving upwards, they started condensing, and these condensation products moved into this chamber now, where they had electrical sparks. So these were the lightning that was going on in the earth's atmosphere. So here there are electricity. And now because you have these gases in the form of condensed material into the in present in the environment, and you have electricity which is giving them energy, they started forming molecules, they started interacting with each other. And again you have these conditions coming, that there is a cyclical flow of increase in temperature and then reduction in temperature. And again the temperature went down, and then they found that all these processes which were going on during the experiment gave rise to the first organic molecule. An organic molecule is what is the constituent of all the living organism, and what they found here was amino acids and proteins and fats, and this is what makes up a body of the organisms. So this very beautifully they demonstrated how life came about to be on earth. So the conclusion here was that they supported the apparent Hellenian theory of origin of life, that organic molecules are created from inorganic molecules during the course of origin of life. And so now on the basis of this experiment, now we can divide the events that took place for the origin of life on Earth in three different categories: chemogeny, biogeny, and cognogeny. Chemogeny is a creation of complex organic molecules from basic chemicals such as polysaccharide fats and polypeptides. Biogeny is the first life which is formed, and cognogeny is the diversification. So they started diversifying that little cell that you just see in the coming slide. They started diversifying and forming all these complex organisms that you see in your environment today. So we'll go over these how like how the chemicals started forming first of all. So as you can see on the leftmost picture, this is the first this is the first step of Earth's formation. The Earth is a boiling mass. You have all these elements: nitrogen, hydrogen, oxygen, carbon present in the environment, but they are not able to react with each other because there's way too hot temperature for them to do anything. So Earth was here a fiery spinning ball of hot gases, and they had dicarbon, cyanogen, and metal carbide. Oxygen here was not present in free state, but they were present in the metals which were there. So the aluminium oxide, boron oxide were present like this. Now, temperature started reducing, and these who were not able to interact with each other, the inorganic components like the carbon and hydrogen, they started interacting with each other, and they started giving rise to something called ammonia, where one nitrogen interacted with three hydrogen. Methane, we have one hydrogen which we interacted with one carbon reacted with four hydrogen. And we have water, where when we have two oxygen molecules bound to a single hydrogen. And then with this we can see that inorganic compounds started. Now remember, inorganic compounds, so these are complex compounds. These are compounds that have started interacting with each other. Now these inorganic compounds gave rise to organic compounds. And now simple organic compounds started undergoing different modes of reactions to give complex organic compounds. So we had already seen that they had formed, for example, here we can see methane, or they can all form ethane. So all these compound now started interacting with all the other, like for example, water started interacting with methane, water started interacting with ethane, and then we have different composition of each, giving rise to complex organic compounds like acetaldehyde, aldol. So the acetaldehyde interacts with another molecule of its own to give rise to aldol. So this was condensation reaction. We had oxidation, oxido reduction. So the hydrogen, so the water started interacting with acetaldehyde to give rise to acetic acid and ethanol, ethyl alcohol. And then you have polymerization. Now these single components started polymerizing with it themselves. So you have these long chain compounds forming. So you can see the complexity of the compounds that are forming is going higher, and you have diversity of organic compounds at the moment in the environment. Now, at this point, you need to note that the sources of energy were UV rays, volcanic eruptions, and electrical energy, and this is exactly what was simulated in the experiment of Miller and Urey. And once here you have this organic compound and you have water also in the which as a part of your hydrosphere, you start polymerizing them. You start forming polymers in the form of amino acids, sugars, glycerol, and fatty acids. So then you have macromolecules formation, which is where you get, so amino acids constitute protein. So you have amino acid formation, then the amino acids come together to give rise to protein. You have sugars, you have glycerol, you have fatty acid. You have sugars coming together to form carbohydrates. You have fatty acids coming together now to form fats. You have now the basic unit that is required for a living organism. You have proteins, you have carbohydrates, and you have fats, the components that constitute a living cells. So you can imagine, if these are present in the environment, there will be a point of time that they will come together and they will start staying together with each other. That's exactly what happens in few steps. So you see the hydrocarbons now, they react to form nitrogen bases, purines and pyrimidines. Purines and pyrimidines means adenine, thymine, adenine, thymine, and guanine and cytosine. Now, if you just go back to the structure of these amino acid nitrogenous bases, you will know there is a sugar, there's a phosphate, and there's a nitrogenous base. You have all these raw materials present now to form DNA, to form a genetic material that can polymerize itself and form an organism. So this at this point, this is also called primordial soup, which was given by Haldane, who said that the oceanic water now had rich blend of organic compound, which are also called hot dilute soup, and these could give rise to the first cell or the first life. So up till now, chemicals have formed which can serve as a raw material for the formation of biomaterial or the living cell. So then we go to biogenic events, where first we have nucleic acid and nucleoproteins. So these nucleic acid that are just from DNA, RNA, they start aggregating with proteins to form nucleoproteins aggregations. And as I said, that they started retaining each other. So due to we have the first cells here, who are also called coercivates. So you see, you have peptides. So these first are just these nucleoproteins together. They are called the first primitive coercivates. So due to intermolecular attraction, the complex organic compounds of primordial soup are collected to form large colloidal cells. Colloidal meaning they are just held to each other by interactions, by forces, like attractive forces, and they are called coercivates. And because they are, so imagine these guys are coming and staying to each other, so they are multiplying by their ability to break and grow. So you break, go and bind to someone else, again break and go and bind to, so you are multiplying yourself in the environment. So these coercivates are called the primary organism. So according to Apparent, these coercivates were the first cellular organism, and they were created 3.8 billion years ago. Just to highlight, Earth was formed around 4.5 to 5 billion years ago, and the first cellular organism, or also called the protobions, were formed at 3.8 billion years ago. And now you have these different proteins. Now remember, proteins are structural components, and their function is a derivative of their structure. Now these proteins, they are folding in different nature. They're folding with different proteins. They're folding with other metals. As and when they started forming different structure, they have different function arising out of it. So you have some proteins which are now acting as enzymes because they will have some metals that can help them to break off a particular compound. So they become a particular hydrolase or oxidoreductase that can break off a particular compound. So like this, you have now enzymes which have started coming up. And the eubionts were identical to protovirus, a present day virus. They were anaerobic and prokaryotic chemoheterotrophs. So what they mean is, so as and when they started enzyme, the what do enzyme do? They break off and they form new complexes. So these are the virus or these are the eubionts, and you have already peptides in the environment. Now you have something which can work as an enzyme with peptide. So now you have both the components, something that can go on its own and does its own reaction, and it also has its own raw material along attached to itself. So it starts getting more complex as and when you start moving ahead. Now, how does the complexity goes? So we begin with, so I'll just go back here. So it said that they are prokaryotic chemoheterotrophs. Now, chemoheterotrophs mean they are using chemicals for use as energy. So what do you see here is the first organism or the coercivates or eobionts, they had electron donor as organic compound because the raw material is organic, and they give out carbon dioxide, and they have oxygen as electron acceptor which give out hydrogen. So they are using the material which is already present in the environment, not synthesizing any of their own. But these chemoheterotrophs, they then evolved to form the chemoautotrophs, which are the nitrifying bacteria, where they started using different source of electron, like different electron donors. They started switching from organic compound to inorganic carbon compound. So from the carbon oxide, they have started producing organic compounds. And for them, the electron acceptor remain oxygen only, but they've now started to giving out organic compounds here. So remember earlier they were the chemoheterotrophs were giving out, or they were respiring carbon dioxide, but now you have autotrophs who are giving organic compounds. Organic compounds are raw materials. That's why they are autotrophs, because they are able to synthesize something which can be used as a raw material by other organisms. Now, chemoautotrophs then went ahead to use photoautotrophs. These people were using chemical energy. These people started using light as a source of energy to carry out these interactions. They use light to produce the organic compounds and water. And then they evolved to become cyanobacteria, who had the first oxygen. Now, here, when you have photosynthesis, remember you have oxygen coming out. Now oxygen has started becoming free in the environment, so the bacteria now started taking oxygen into their system and then started producing organic food using that. So these are the first bacteria who started becoming oxygenic in nature. So they started using free oxygen to synthesize food. So they are called oxygenic and aerobic photoautotrophs. And you see the timeline, this is around 2700 million years ago. So we have covered from 4.5 billion years ago till some 2700 million years of evolutionary history. And then once you have cyanobacteria, these are prokaryotes. Now you have these prokaryotes starting to become eukaryotes. How does that happen? You have photosynthetic prokaryotes. They start using aerobic respiration, because I just told you that they have started using oxygen now. So the cyanobacteria developed a true nucleus over the period of time, like prokaryotes, and transformed into eukaryotes. But they were still unicellular. So these are one single cell. Now imagine these cells coming together to form multicellular organism via colonization process. And multicellularity is the step where you transform to a tissue level so a tissue level of expression. So basically what you mean, the cell was doing one function, now you have multiple cells who can do the same function. So you, very crudely, if I say there are 10 cells coming together, you have 3 cells doing one function, 2 cells doing some other functions, you have 2 cells. So you have functional repertoire also increases, and you can do multiple functions now. So that's how complexity arises from the simple constituents. Now, all this makes sense if you have some evidence to show it. Like you need to prove it, and I already mentioned in the previous slide as well that you need some evidence to give to back your hypothesis. So, and how does that happen? So there are morphological evidences, there are embryological evidence, and paleontological evidences that support that organisms have come from pre-existing organisms. And how did pre-existing organism arrive? That we have covered very well by the Miller and Urey experiment. So now what are the morphological and structural evidence? We say that organism came from previous organism, and we also mentioned that complexity increases over the period of time during evolution. And you can see that the body organization keeps getting as more complex as in when you pass time. So you start with a unicellular amoeba, move to a multicellular sponge. The multicellular sponge now demarcate different functions to different cells, and they become tissue level organization. And then you have tissue level organization, the tissues coming together to form several other perform several functions. They become organs, for example in fishes. And then you have so these are morphological structural evidences. So first is body, the second is homologous. How do you remember homologous? Homologous means they have similar structure but different function. Why different function? Because they are all in different environment, and they will use that particular part of their body for some other purpose that is amenable to their environment. So, for example, if you see the four limbs of animals like cats, human, whale, and bat, you see them all having similar structure. What is the similarity here? The pentadactyl nature. Pentadactyl means five fingers. So, five fingers here, five fingers of cat, five fingers of whale and bat. So, all they have similar structure, but the functionality differs, because for human, this is just our hand that we just use for, in very crude sense, if you just gonna pick something or hold something. But cat uses the same for movement. Whale uses that for swimming, and bat use them for flying or gliding. So you have different functions, but the structure component remains the same. If they are structurally same, that means they have arisen from the similar common ancestor. And for plants, you have thorns and tendrils. So thorns in Bougainvillea and cucurbita, in cucurbita you have tendrils. They have different function, but they are the same structurally. They are outgrowth from the stalk. So they are outgrowth from the stalk, but one is used as a stationary purpose, and the other is helping the plant to grow. So you have similar structure but different function as homologous. But the converse, where you have similar function but different structure, is also there, which is called the analogous organs. Why will you have similar function? Because you are staying in the same environment, or you're using this structure for the same function. So if you see the wings, they are all they are all used for the same function, but structurally they are very different. So because they're functionally same, they are called analogous organs. So you have bat wing. Look at the structure of all these wings, they are so different. One was pentadactyl, the other has feathers, and the others has just two parts which are supported by a midrib. This was for wings. You can also see this for fins and flippers. For example, fins in shark and flippers in dolphin are used for swimming, but they are structurally very different. So this is why they are analogous organs. And another example of growing complexity is the heart, or what we call gradual modification. So this is a snapshot from Pisces for vertebrata who have complete circulation. Now you see how the heart chambers have evolved. Pisces started with just one auricle and one ventricle. Amphibia has one ventricle, and the structure also changes, and they have two auricles. We move to reptiles. They have ventricles that is also divided into right and left, and auricles which are again right and left. But you see this septum here which divides these left and right, they're incomplete. But if you see in birds and mammals, you have this complete septum. If you have drawn heart figures, you would know that there's a complete septum here which divides your right and the left portion of your heart. So that's how gradually these organs have modified themselves. And for this to happen, there has to be enough time, one, and they have to evolve complexity at a linear level. So first auricle divided, then the ventricle divided, then the septum came, and then the septum went ahead. So you have to give them time to evolve this complexity. And then you have things like this, which are called connecting links, which have characteristics of two different classes or two different class levels, and they can be connecting link because they will be the ones that can demarcate that part of this is reptile, part of this is some other organism. For example, here you can take amphibians, and then the following organisms which have only amphibian characteristics will be the one that carry that particular characteristics. For example, let's say you have something which can live on land and water, and then you have that particular organism is able to climb as well. Now it has characteristics of being on land and dual habitat as amphibians, but it can climb and glide also, so it will have reptile locomotory organs. So now you have some organism which have these locomotory organs, so you classify them as reptiles, and you have some organism which live on land and water, and you classify them as amphibians. So they give you this connection between these two classes and how they evolve from each other. So we have several such connecting links. We have euglena, who are like plant and animals. We have peripatus, who are between arthropods and annelids. We have duck-billed platypus, who are between reptiles and egg-laying mammals. Now, reptiles and mammals have a bird in between, but these are connecting link between reptiles and egg-laying mammals. We have chimera, who are between the cartilaginous and bony fishes, oxycthyre and scondycthyres. We have coelacanths, who are between bony fishes and amphibians, so pisces and amphibians as a class. And then you have spinodon, which are amphibians and reptiles. So all these are structural evidences. You have embryological evidence as well. So if you see the development of embryo across several classes, for example, fish embryo or salamander embryo, tortoise, chick, rabbit, and man, you will see the stages are similar, and the morphology of the stages or at the stages how the embryo looks is also very similar. And this can only be if they have all evolved from one another. They have this structural component that is similar to like common to each other, and that's why they are all evolving from each other. And then you have fossils as an evidence. So for example, you have horse. Horse evolution is very well documented because you have fossil evidences from different stages of geological time scale. So you have used from Eocene period to the recent uh, epoch. You have fossil evidences that show the complexity and size of their skulls, of their bones, of their sizes, and that gives you an idea how with time there is a linear evolution going on. Now, I said linear evolution, or I wanted to mention about how evolution happens. So, all these are evolving, yes, but how are they evolving? That is also something you should know, that how we see all these evolution taking place, but how does this evolution actually happen? So, you must have read about theory of inheritance, which was given by Lamarckism, and who said that the evolution is because of change through use and disuse, and organisms driven to greater complexity, and you acquire some characteristics and you inherit as it is, and this is because of the environment that you are surviving in. And he gave giraffe as an example. So he said, as and when the tree started growing higher, the giraffe started using their necks more and more to you eat the or to reach to the leaves. So their necks also started growing up or growing larger, and that's why now you have giraffe who has a very tall leg. Or something like flightless birds, who he said that they started losing their like this they so on the contrary, they said that these flightless birds, they started losing their flight mode of wings because they had no more use of it. So because we also say use and disuse. So because it was not used anymore in their environment, they started like losing their wings as a matter of fact, and they became flightless. But you have criticisms here. The first thing is, he said that the complexity always increases, and you always see them increasing to larger. Okay? But you have examples like trees, and you have example like shrubs, and you also see it by evolutionary method that these shrubs came later than the tree. How did this happen? If you want individuals to grow larger, shrubs are shorter than trees. How did that happen to be? So that was the first evidence that could refute this theory. Second was, he said that you have inheritance of acquired characters. So now you have this man who has his one hand chopped off. Now ideally, according to Lamarck, if he is to reproduce, he will have a baby who will also have only one arm, but he has a baby who is born with both the arms. So that this also refutes its second part, second conjecture. And the scientific evidence or the experimental evidence for this disproving this point was germplasm continuity or Wisman's theory. So what he did was he had these mice. Just a second. Yeah. So he had these mice, and he chopped off or he cut the tail of all these mice and allowed them to breed. Now, if you chop off the mice tail, you will expect the new mice to not have tail. But in every generation that he did this experiment for 22 generation, and in each generation he found that the young mice will always have tail. So he said that it is not the physical characteristic, but there is something in the germ plasm, or which is something in the blood, which is going ahead in the continuity of these traits in the organisms. Now, this was further taken up by natural selection or Charles Darwin, who proposed a theory of natural selection. And he said that these diversity is because of rapid multiplication, where you see population increasing geometrically. And then there are limited food resources and space availability. So each organism adapts to its environment, and population size remains constant because of lack of food, but they diversify because they gain new adaptation to adjust to their environment because of the struggle for existence. And then he said that this is not because of use and disuse, but these are because of variation which are already present in the population that get selected to continue or propagate in the environment. And there is natural selection where they have the survival of fittest. So all these uh, organisms, or in this, for example, there's are finches. So all these finches have survived so far because they had these variations that helped them to adjust to the new environment or take up a new characteristics, and they could survive in that environment. So, and then these characters are, because they are successful to them, they are also inherited in their offsprings. So you have inheritance of the useful variations, and then because of this, because of these new adaptations only, you have new species. But to understand that further and to understand the variation, which he said that the variations are present every time, which was further clarified by Hugo de Vries in his Enothera experiment. He said that new species, unlike what Darwin said, that their variations were present already and you use those variations to survive to in a new environment. He said that new species arise from the pre-existing one, but in a single generation using a sudden appearance of mutation. So you have something mutating, which gives you a different characteristic, and if that characteristic is beneficial to you, you propagate them further in your lineage. And that's why he said evolution is not continuous, but it is discontinuous and a jerky process. And this mutation is what is the raw material of evolution, not the variation. So this is what I expect that you would have covered in your theory classes. So now I will go over some previous year's assignments so that you can solve them based on how what you have just gone over and what you have studied so far. So you have 4 questions here. I'll read out the questions and I'll wait for maybe 10 seconds, and then I will show you the correct answer. And if you have any doubt, let me know. So the first is the father of microscopy, who was either Anton van Leeuwenhoek or Zacharias Jensen, Pasteur, or William Harvey. The second question says the organism lives in extremely salty areas, and they are called Halobacterium, Thermoplasma, Pyrococcus, and Sulfolopus. The third is smallest unit in taxonomic classification with fundamental similarities is. So you have to imagine the taxonomic rank pyramid here. So it's either family, phylum, genus, or species. And then you have the fourth question being the phylum that is closely related to both chordates and non-chordates and is considered as a connecting link between them. So again, if you just imagine the classification system and you try to see what comes between the chordata and the previous phylums. So these are the answers. I hope you got them right. So I'll give you a cue to how to do these especially. So if you want to do this, you see the terms. The terms only where they reside is located. So halobacterium, halo means salt. Thermoplasma will mean something in very high temperature. Pyrococcus will means again in something in very high temperature, but this is a different shaped organism. And sulfolobus in sulfur containing environment. So in an environment which has high sulfur component, it is the organism that lives in that environment. So you can guess these kind of questions with the terminology as well. And here, as I had given you the mnemonic of family, keep ponds clean or else frog gets sick. That sick is the smallest unit. So you remember it using that mnemonic or any mnemonic that you want. But species is the smallest unit. And hemichordates is between the protochordates and the or the chordata and the non-chordates. And then, so we move on to the next set of questions, which is the ribosomal RNA present in both prokaryotic and eukaryotic ribosome. I did not do this because I did not cover in part of my syllabus, but I believe this was covered in the theory. So it is 5S RNA. Then you have the protein-rich layer in cell body of euglena. Euglena is protista. So again, because I did not mention this in the presentation, these have pellicle layer which give them the motility. This I had mentioned, the type of association where two organisms function as a single unit and help each other for survival. So here if you come to the other terms also, they are equally important. So symbiosis is where I said that this is a functional, beneficial interaction. But asymbiosis means the opposite, that there is no beneficial interaction. And then you have the classification for that kind of interaction where you have emancilism and neutralism. Emancilism is when one is not gaining any advantage, but the other is actually gaining disadvantage. So for example, you have something like parasite, but not a parasite would be not a proper example, but something which is living just as an accessory to an organism, but leaching off the components from or the nutrients from the body. Now it is not doing, the one which has lash is not doing for any beneficial purpose. It is just there because of chance. But from the organism or from the host where it is leaching out nutrients, it is doing harm to the host. So that is an emancilism kind of interaction. And then you have neutralism where there is no benefit or no disadvantage to either of the interacting partner. And then it comes to symmetry. So we had discussed symmetry radial, bilateral, and spherical. So given the body type, which one of these phylum will have a radial symmetry? So if you imagine Porifera was sponges, Cnidaria and Annelida had segments, and Platyhelminths. So the answer, because it is radial symmetry, it is Cnidaria. Then you have next set of questions which said compound eyes are present in octopus, mosquito, frogs, and humans. Then we have question which said common method of asexual reproduction in Hydra is conjugation, spore formation, binary diffusion, and budding. Then mollusks use their feeding organ called radula to scrape off algae from their rock. The radula is made up of what component? And the correct characteristic feature of heart and blood circulation in mammalia. So just imagine the heart chamber evolution, and which was the last one. So was it an incomplete 3-chambered and double circulation, complete 2-chambered and single circulation, incomplete 4-chambered but double circulation, or complete 4-chambered and double circulation? So I hope you got that right. So so the compound eyes in mosquitoes are actually form of obertida, which helps them look to the peripheral vision. All these four are actually method of asexual reproduction. Binary fission you will find in amoeba or pseudomonas. Spore formation you can find in fungus. Conjugation you can find in bacteria. And budding you can find in bacteria and hydra as well. Now bacteria and hydra are two different kingdoms, but they have used their similar reproduction method for survival or growth. And then you have mollusk. Now you have chitin, which is present in the radula of mollusk, and which they can use to feed off scrap algae. Now, if you just imagine how would that have happened if you didn't know the answer, To scrape off something from the surface, you would need something which is very strong in nature. It is chitin, which is also present in arthropods, and mollusks are closer to arthropods, so you could have guessed chitin from there. And as I mentioned that for the other question, you just need to remember the evolution of heart chamber. And if you just go from for the last image from that where we have seen mammalia, we had seen complete septum, we had seen right auricle, left right ventricle, and left ventricle. So we had four chambered and we had double circulation. And then we have this questions. So we say the type of organization found in grantia that helps in the flow of water through the organism. Then you have a flowerless plant with no true roots but rhizoids as produces spores in small capsule, where there are grass, mosses, ferns, and maize. The first question has these different types, which are different types of sponges that are present in the environment, and I believe that would have been covered in the theory classes. Then you have the coiled tubular duct-like organs which filter and remove waste from earthworm. So you have these different structures, and you need to know which one is used for excretion. And then you have an egg-laying mammal with a beak-like bird. So which you can guess from if you know the if you just imagine the organism and which type of organism it is. So I have shown the answers. So I'll start with the last question. It says a mammal. So king cobra is a snake. Kangaroo is a mammal, yes. But does it have a beak-like butt? No. What is the identifying creature of kangaroo? It has that pouch where it can carry its offspring. But it doesn't have a beak like a bird. And duck doesn't have a beak like a bird because duck has its own beak, which is like a characteristic of its own. But echidna is an egg-laying mammal. Duck doesn't lay egg. In the sense, duck doesn't lay egg and doesn't have a beak like a bird, but echidna is the animal which will have a beak, and which is also the monotreme. They are also called monotremes, which they are egg-laying mammals. And yeah, so the earthworms or the annelids, they have nephridia that they use for excretion. So here, if you see protonephridia, remember protonephridia is even basal to the advanced structure. So nephridia is advanced. Protonephridia is found in organism which precedes the annelid classes or phylums, subphylums. So nephridia starts at annelid stage, and you will find them in cockroaches also. So these are the organisms which are used for excretion. And remaining gonadial and pericardial. Pericardial. The cardial gives you a idea that this is something to do with heart, so it has something to do with circulatory system. And gonadial is used in the reproductive system because gonads gives you the idea. So you should have thought about nephridia and protonephridia, and you would have known that annelids are or earthworms are advanced organisms, so they will have nephridia. Now here you have flowerless plant with no true roots but rhizoids and produce spores in small capsules. So you have grass, ferns, and maize. You needed to know how are they classified. So remember, I told that do they have a proper body? If they have a proper body, proper root, shoot, leaf, they are ferns. If they don't have proper root, shoot, and leaf, then they are algae or they are mosses. So you need to know what did they say. They said no true roots, but they had root-like structure. So then they were mosses. And here, these are the different kind of sponges, ascon, cyclone. How are they flowing the water? So, it's an example of cyclone where you have these different pores that are lined up linearly that can be used for flow of water. And then the next set of questions are the type of scales present on Bombay duckfish are placoid, cycloid scales, or a combination of placoid tenoid, or cycloid and tenoid and placoid cells. And then what is the field of science that deals with the human behavior in context to mental processes? Then the two pair of limbs in the frog, they arise from pectoral and ventral. pelvic and dorsal, only pectoral, or pectoral and pelvic. And then the book that explains how species evolved over time and gradually diverged from each other through a process called natural selection. So these are the four names which are, and you have to select the one which is published by the person who gave the term for natural selection. So I'll start with this. So Darwin is analogous to origin of species, and it says natural selection means it is about Darwin. So this was easier. And then you go to frog. Now you remember where do you see these limbs coming from, and where in the body of frog, where are these limbs located? It is the place where they have arisen from. So you have pectoral and ventral. So ventral is the lower belly part. So the limbs are not from ventral. Dorsal is from back. It is not there as well. But they have two set of limbs and at two different positions. So they have pectoral and pelvic. Pectoral is around the chest, and pelvic is around the mid body, or at the junction of limbs and the back. And then you have a simple question which says field of sight, where you have human behavior. And the other options are very easy. They are history, public administration, and linguistics. They have got nothing to do with any human behavior whatsoever. And then you have scales which are present in forbidden. So these are different kind of scales. The basic unit structure of how these scales are, they define these fishes, or they define the organisms. Then you have these questions, which is which theory is similar to stating God is the creator of earth and organism? The part of earth with large quantity of iron and nickel. So lithosphere is the outermost space. Then you have pyrosphere. Then you have barysphere is the inner core, which also is divided into further inner core and outer core. Or if you have to remember it, it's the mantle, like crust, core, and mantle. And then you have the primitive earth, when cooled down below 1000 degrees centigrade, first led to the formation of what of this? A biosphere, hydrosphere, ecosphere, or magnetosphere. And the reasons for the presence of hydrogen and hydrogen compound with water to remain within the atmosphere of primitive earth is which force? Gravitational, reducing environment, oxidizing environment, or nuclear force? So the first one was easy. Is either special creation or it is the catastrophism theory, where they just mentioned that this is just one single entity who has defined all these organisms on the planet. You have iron and nickel. So iron and nickel are part of which are composed of which part of the earth. So they are part of barysphere. They are part of inner core. And remember, when you look at earth, the inner core it has magnetic properties, and iron and nickel also have magnetic properties. So they are present in the inner core, or the inner core of barysphere. And then I had mentioned this point that if there is reduction in temperature on Earth, what would have it would have led to the formation of. So biosphere is where what you call as the uh, organism, the layer of Earth which is all the organisms. So it is we are talking about primitive Earth, and it first cooled down to 1000 degrees centigrade. You know that it was not possible for biosphere to form at that point. Ecosphere is also related to biosphere, when you take into take into account the organism and their habitat. That is called ecosphere. That is also not possible. Magnetosphere is what I just explained for barysphere. Now, barysphere or magnetosphere was already present in the Earth's landmass. Hydrosphere was what formed after it reduced the temperature, because all those hot gases started condensing and pouring down on the Earth's surface. And here, so all these components, the hydrogen and the hydrogen components with water, remain in the atmosphere because of gravitational force. Because for nuclear force and other force, you first have to have evolution of other complex organism which will go into these environments. So reducing environment, oxidizing, for oxidizing, you need free oxygen, which came when there were first, cytogenic bacteria or oxygenic photosynthesis, reducing environment when you have these organisms releasing hydrogen into the environment. So it's for in the initial period when when these compounds were in the Earth environment, they were held together by gravitational forces. And then you have this question, which is also easy because we did go over this. The first is to do disprove the theory of spontaneous generation. Louis Pasteur, with the experiment involving S-shaped flask, had given the following analysis: appearance of microorganism in the neck of the tube with condensed water. Disappearance of microorganism in the neck of the tube with condensed water. Appearance of microorganism inside the flask with condensed water in S-shaped tube, and no growth in the flask after breaking S tube. And the other question is the correct assumption for the spontaneous formation of monomeric organic compound according to chemical theory is complex molecules interact with each other to form simple monomeric organic compound. Reactions occur in oxidizing environment inside the ocean. The organic compounds and simple organic compounds were present in the atmosphere. And as the temperature of the earth is lower, the inorganic molecules were condensed in the form of rain. So just take a while, think about these lines and try to guess the answer. I hope you got that right. So the first one said, which one was the analysis that disproved the theory of spontaneous generation? So we'll start with the incorrect answer and why they are incorrect. So you have the option saying no growth in the flask after breaking S tube, which was wrong, because if you break the S tube, you bring into contact the air with the flask, so you get growth here. Then you have appearance of microorganisms inside the flask with a condensed water in S-shaped tube. So you have the tube intact, and but the organism is inside the flask. So if you have condensed water there, but how come there is organism in the flask? If there is tube present, there would be no contact between the air and the flask contents. This is appearance of microorganism in the neck of the tube with condensed water. So if there is in the neck, if there is condensed water, and if there is environment, if there is contamination, I mean, if there is condensed water in the neck, then you will have an organism appearing there because it has the environment to grow there. So that's why the first option, which said that there is an appearance of microorganism in the neck of the tube with condensed water, is what Louis Pasteur observed. And for the second question, where we said that how did these monomeric organic compounds spontaneously form? So the first option says complex molecules interact with each other to form simple monomeric. This is the reverse. We went over this, that simple gives rise to complex organism and not the opposite. Then the reaction occurs in oxidizing environment inside the ocean, which is also wrong because oxidizing environment came very later when the cyanobacteria started forming the or started using up oxygen for their air respiration. And this is here we are talking about monomeric organic. This is way ahead before the nucleoproteins were built up. Third says the organic compounds and simple organic compounds were present in the atmosphere. This also we know that this is not, there was not something this was present, but these were all formed in due course of time. So that's why the fourth option, where they say that as the temperature of the earth is lowered, the inorganic molecules were condensed. This is what we repeated in the previous slide as well. That as the temperature started reducing, the hot gases started condensing and came poured down as rain to give rise to hydrosphere. And then, okay, this one. So we have the first question is a theory strongly supported by Arrhenius for life on Earth is steady state theory, theory of panspermia, spontaneous generation, and cosmozoic. Then the gas responsible for life in the current Earth's atmosphere, but it was not allowing the condensation reaction in primitive Earth. So you have to compare today's environment and primitive Earth's environment. Then the mixture of gases in the exact ratio by Miller and Urey, who had used ammonia, hydrogen, and methane, but in exactly which composition or which ratio. And then you have the chemical theory. According to that, the energy for the formation of inorganic molecule was provided by which of these sources? So I hope you got that right. So the first one was Arrhenius. If you remember, Arrhenius and Richter were supporter of theory of panspermia or cosmozoic theory, or Arrhenius was strongly supporting the presence of spores from the meteorites that would give rise to all the microorganisms and the diversity on the planet. And then we have talked about this, the gas responsible for life in the current atmosphere, because remember, oxygen is what is thriving and helps you grow. But this is not in the primitive environment, so oxygen. And then I had mentioned about 2 is to 2 is to 1. So you have methane, ammonia, and hydrogen in the ratio of 2 is to 2 is to 1 in the Miller and Urey experiment. And this was also mentioned because if you just remember the Miller Urey experiment, in the last chamber, they had provided lightning as a source of energy. So ocean was not the source of energy. Plant came very later, and electricity was not. Electricity came because of lightning. Electricity was not so like present as itself. So, okay, the next part is the era which has no available fossil form: Cenozoic, Mesozoic, Paleozoic, or Archeozoic. From the following examples, which characteristics do not support the theory of inheritance? Theory of inheritance by Lamarck. Loss of vision in case of mammals, cave animals, development of webbed feet in duck, development of legs in lizards, and well-developed pinna muscles in rabbits. Then, what is the modified pentadactyl forelimb in dolphin called as and is based on the category of which evolution? Remember the examples of dolphins and compare it with the fins and the flipper example that we went over in the discussions. And the effect of radiation and chemical on germ cell is used in support of which theory? So try to remember, where did they use germs? Where did you remember the germ theory, and why was that used for disproving which theory? How this is the answer that you got. So, so the era Archeozoic, as it says, Arche is very primitive. So there are no fossil for Archeozoic because there was no condition available for that period. One, second, you need some solid structure to be preserved in fossil form. And Archeozoic was considered like consists of all prokaryotes region, prokaryote organisms. So they were not hard shell that they could be retained in fossil forms. So that's why Archeozoic has no fossil available. Right? We are now looking at characteristics which do not support the theory of inheritance acquired characters. So we'll go over each and see how are they supporting the theory. The first is loss of vision in case of cave animals. Lamarck said there is disuse and use of organisms or organs. So in cave animals, you do not get to see anything because it's dark there. So that's why you have loss of vision, which supports what Lamarck is thinking, what the line of thought for Lamarck. Then you have development of web feet in ducks. These ducks are swimming in water. So web feet will give you more space to swim easily. So again, use. So use a disease theory. You have well-developed pinna muscle in rabbits because they use this for sensing. So again, yeah, use. But development of legs in lizards. Lizards, you find them climbing. So why will you have legs and not webbed toes? Is why you say that this does not support the theory of inheritance. And then you have the question where you are supposed to see, compare the first of all you need to know what are the dolphins forelimbs called, and where you had find this example. So they are called flippers. And then how did we discuss them as part of divergent evolution or convergent? Convergent, remember, if you get confused between them, divergent means diversifying. What diversifies? Function diversifies. So when did we read this? We said structurally same but functionally different. So if you have different functions, you are diverse. So that's why it's a divergent evolution. but you are structurally different, but you're converging at the point of function. You have similar function, so you are convergent. This is convergent evolution, where you are converging on the function. Now, where did we read the flipper example? In flippers and fins, where we said that both of them are used for swimming, but the shark fins are different in structure than dolphin flippers, so they are converging at functional level, so they are convergent evolution. And the last was the effect of radiation and chemicals on germ cell is used in the support of which theory? So, theory of natural selection, Weismann's theory of continuity of germplasm, Hugo de Vries of theory of inheritance of acquired characters. So, you when you say germ cells are used in which theory, it is in the support of which theory, or in the where does it say? So it says it. Had seen this for Lamarck's example, where he said that if there is chemicals and radiation acting on a particular organism, it will affect the cells in such a way that the coming up or generation also possess the same characteristic or the same like problems that they have inherited. Okay, the next set of questions are: who disproved the theory of inheritance by acquired characters? And we have then, which among the following metal is used to determine the age of fossils? What is the connecting link between cartilaginous and bony fishes? And what is the principal factor for natural selection as recognized by Darwin and Wallace? So the first question said who disproved the theory of acquired characters. So we go back to the experiment of the tail cutting of mice by August Weismann, who said that it is not the characters in the it's not the acquired characters, but it is the germplasm theory. It is in the germplasm. It is the blood that flows the flows through that carries the information for the next generation. And then you have question where it says which metal is used to determine the age of fossils. It is potassium, because you don't use sodium and iron. And then the connecting link between cartilaginous and bony fish is chimera. And we had all gone over the others as well, Coelocanthus sphenodon. And Darwin and Wallace. So it says what is the principal factor for natural selection? Variation is present in the organism, but variation comes into play when there is a change in the environment. So environment is the natural principal factor for Darwin's and Wallace's theory. And why Wallace? Because Darwin came to the theory of natural selection based on the observation made by Wallace and Malthus. So both these people, they had seen several observations in their field experiments, and those observations helped Darwin to form the natural selection theory. So another important point is Darwin formed his theory not only on the observation of finches, but also because he knew Malthus and Wallace's observations. And then we have this. So we say what led Charles Darwin to propose a theory of natural selection in his book of Origin of Species. I just gave the answers, so if you just go over the options, you would be able to understand that. And which of the following experiment is false regarding the appearance of mutation in offspring in Hugo de Vries' Enothera experiment? So if the traits are constant and plants are stable. So plants means because he was using Ornithera, so he said they are stable. They are retrogressive with lost traits, they are progressive with newer traits, and they are digressive with weak and low survival. So the first one was easier because I had just explained this. The second one said which one is false for the mutation theory. Remember, Bryce said that mutation arises and that is the raw material for evolution. Now, the second is retrogressive with lost trait. So you can lose a trait due to mutation. Third says you can gain a new trait with mutation, and both of these are possible because mutation doesn't have a specification or a direction. See, because of a mutation, you can lose a trait. Because of a mutation, you can gain a trait. Because of a mutation, you can have low survival as well because that mutation doesn't know which way to go. But if they are stable, that's the first point which says constants and plants are stable, then you have no opportunity to have mutations and then you do not evolve. And that's not what we observe. So that's why the first option is wrong when you think from the Bryce experiment. So these were the questions that were present in previous years' assignments. So for the next doubt session also, I will try to come up with some questions on my own and some previous year assignments so that you are able to understand the topics better and you can solve them.